Welcome to the first lecture of microbial metabolism, and in this lecture we're going to be looking at an introduction to metabolism, and I'm only going to be going over these things quite briefly, okay, so don't get too worried about the different parts of it. So basically metabolism, every cell needs to undergo some sort of metabolism to produce food, okay, to produce energy. And this is, part of this is the oxidation and reduction reactions. So that just means that electrons or ions move from one thing to another, basically. And we're going to look at aerobic respiration, where you get the generation of ATP and how some of those things actually occur. So a bit of stepwise going through it. And then the chemiosmosis as well. So these are the things, these learning outcomes are the areas that you need to be able to answer fully. Okay? So explain, discuss, etc. So these are probably good ways to do your after lecture notes, so you know what you were listening to. So just in brief, metabolism is just a series of events, series of reactions. So one happens, then the next, then the next, then the next, okay? So it's kind of stepwise. And then there's two main types. You've got the building up, so the anabolism. So if you need to if you've got simple structures or substances, you can build them up into more complex structures. Okay, so that's anabolism, and usually it requires energy. So things like growth or reproduction, repair, all require energy. So it's and building up. So that's the anabolism. But if you've got a more complex substance and you want to break it down to release energy, then you've got the catabolism. So it's breaking down. And things like movement, you need energy. So you get breakdown of, say, glucose, for example. Breakdown of more complex organic molecules into simpler substances. Okay? And this is quite a nice little diagram just showing you the difference. So you've got the larger molecules, when they're being broken down into smaller molecules, that's catabolism, or if they're being built up, it's anabolism. Okay? But there's always that energy requirement as well, either giving or taking. So if you look just generally, there's two different sorts of energy that are available. So we've got both sunlight and the redox reactions. And the energy for chemical reactions is simplistically stored in ANP. And so ANP is adenosine triphosphate, so there's 3 phosphates, and all organisms actually produce ATP. And so what that actually looks like is you've got adenosine and the 3 phosphates there, okay? So if you broke down ATP and released that energy, then you would have ADP, so you've got diphosphate, and you've got energy. Okay, so that's just how the energy is released. Now the redox reactions are basically coupled reactions. So you've got one molecule loses or one or more electrons, while the other molecule gains those electrons. Okay, so you've got the oxidation, which is losing one or more electrons, and reduction, which is gaining one or more electrons. And these happen at the same time, because if it comes off one, it's got to go somewhere. Okay, and if you look at those electron transfers, they that transfer of electrons, they basically drive cellular metabolism. Okay, so, and it's not always oxygen that is included. Sometimes you can have these processes occurring in an anaerobic environment as well. And this is actually a really nice table to compare oxidation and reduction. And so you've got the oxidation here, so gain of oxygen, loss of hydrogen with oxidation, loss of energy, exothermic. Okay, so exothermic or exergonic means that it gives off energy, gives off heat energy specifically. So if you add two chemicals together, if it's an oxidation reaction, then it typically, that solution or whatever, will become warm. It gives off heat. With the reduction reaction, typically it requires energy, so it gets quite cool. So pretty interesting. Okay, so gain of with oxidation, gain of oxygen, loss of hydrogen. Everyone happy with that? Yep. So now we come to the fun part. So don't get too worried about this. This is just showing you how electrons actually move through the system, okay, to allow that production of energy. And for that to occur, you need that production of energy, you need to have carrier molecules to allow it to occur. So it's, if you look at it a little bit stepwise, okay, so basically, if we just look at this first part on the left-hand side, you've got an electron coming into the system, and it's causing reduction, which means what? Gain of electron. So this is less positive. Yep. Then it undergoes oxidation, which means it loses an electron, which means it gets more positive. Yeah, that's all it means. And we've got a couple of different substances or substrates in our body, so FAD and NAD, and that can have hydrogen with it or not, and they aid in that cycling of electrons. That's all it is, okay? And that cycling of electrons helps with producing energy. So usually if we've got ADP, adenosine diphosphate, if we add another phosphate, it goes to ATP, where it's storing energy. So that extra phosphate is what's used. But so the addition of a phosphate group is known as phosphorylation. Okay, so that's just a bit of a term for you. And there's three mechanisms for phosphorylation in the body. So you've got photophosphorylation, substrate level, and oxidative phosphorylations. Okay, so basically it's the phosphorylation, so you've got the ATP being produced, and that produces the energy or provides the energy. So if we start with the first one, we've got photophosphorylation. So just the name actually should indicate where the energy is coming, where the light energy is coming from. So it is a light energy. If it hits chlorophyll, remember what chlorophyll looks like? Yes? Then it excites that, and you get electrons movement of electrons, and you get ATP being produced. And once it comes down to a lower energy level, it's already released its energy, the ATP, and so it comes down to that lower energy level, ready to be photophosphorylated again. Okay, so it's just, once again, a cycle. Then we've got the substrate level phosphorylation. So this is If you think of this big blue thing, big purple thing as an enzyme, okay, so what do enzymes do? They catalyze reactions. Yeah? So you've got a phosphate group here, and it actually joins to another phosphate group on adenosine. So you've got adenosine diphosphate. Now what happens there is when that joins, you get the production of ATP. So you get the phosphorylation occurring here. You get generation of ATP, and then you've got this lone phosphate that that phosphate has been taken off the organic molecule, and then you're just left with the remainder there. Okay, one phosphate has been taken to produce the ATP. So that's all it is. So it's a direct transfer of a phosphate group, so this one, to ADP for the production of ATP. Then we've got oxidative phosphorylation. Now this is a little bit more tricky. So you've got energy released as electrons, and you've got a series of events that occur and then that end electron is used as in the production of energy. And so for this you need some of those coenzymes that we saw before. So the NAD and the FAD and the NADP. Okay, so you've seen those before. They just are part of that cycle. And not only are we transferring electrons, but we're moving protons as well. Okay, so that looks like, and I know it looks pretty messy, but just bear with me for a minute. So if we look here first at the electrons, okay, we've got electron movement and a couple of different steps. So there's about 4 complexes that it moves through, okay? And at each of these steps, the hydrogen ions, so we've got channels here as well. So the hydrogen ions, intracellular hydrogen ions, are actually pumped out to the outside by a hydrogen pump. Okay, so we've got movement of electrons, but also movement of protons. And you can see that there's a lot of movement of protons. Okay, and then we get this electron coming back, and the recipient is oxygen, and so when that occurs, it causes this ATP synthase complex to be activated, and the hydrogen ions or the protons come back in and cause that ADP to join with another phosphate to form ATP. Okay, so it's just movement of electrons and hydrogen ions. We've got a number of different pictures of this, so we'll just revisit it. But it's just so you're aware of the hydrogen ions being pumped out and then coming back in across that membrane. Happy enough? Yes, okay. So the processes that use the 3 mechanisms for generation of ATP, we've got cellular respiration, we've got fermentation and photosynthesis. Okay. The cellular respiration, as I said, could be aerobic or anaerobic, so using oxygen or without oxygen. And the fermentation, so you've got oxidation of those organic molecules, and then ATP production by that substrate level phosphorylation. And then photosynthesis, so we've got that light energy, and they can be cyclic or non-cyclic reactions as well. But we need to also think about energy for the microorganisms and capturing or obtaining carbon. So there's another couple of terms that you need to know. Autotrophy, which is self-feeding, so auto is self. So self-feeding, and heterotrophy, which is other feeding. Okay, so just the 2 terms. So we'll have a look at these different, so actually if we just look at the names, photoautotrophs, what's involved? Light. Chemoautotrophs? Chemicals, yeah. And the same with the heterotrophs, okay? But if we look at this in a bit of a table, we've got, say, all of the microorganisms, and you've got the autotrophs on the left, the heterotrophs on the right, and you can see if we go along the bottom from left to right, we've got the photoautotrophs, the energy source is light, and you've got some you've got some examples there. And then the chemoautotrophs, you've got some the energy sources there which are inorganic compounds. So you've got some of the so sulfur producing bacteria, etc. Okay, using. And then if we go to the heterotrophs, you've got some examples of the photoheterotrophs and the chemoheterotrophs as well. Okay, so you can see with the chemoheterotrophs, these are the ones that we generally come in contact with in microbiology. Sometimes you might look at, have a look at the other ones as well, but mainly it's the chemoheterotrophs. And this is just a distillation of that last slide, looking at the how they live, and the chemoheterotrophs, the disease-causing ones, these are the ones that get their carbon from an organic source. Okay. And if you look at photosynthesis, so what does that actually do? You've got CO2 and water, and if you've got light and chlorophyll, then you can get the production of glucose and oxygen. Yep. In photoautotrophs and chemoheterotrophs, using aerobic respiration, you get glucose plus oxygen, because it's aerobic. And what does that give you? CO2, water, and ATP. Pretty simple. Now, if we look at that in combination, you've got the photosynthesis and respiration. So you've got the, when we've got photosynthesis, you get the production of glucose and oxygen. Glucose and oxygen are used for respiration by humans, for example, to produce CO2 and water, which are used in photosynthesis to produce, okay, so you've got that cycling effect. But respiration itself, so cellular respiration, we know about respiration. When we breathe in, we breathe out. That's respiration. But cellular respiration is what happens in the cell itself. And so in aerobic respiration, oxygen is the final electron acceptor. And so you've got glucose plus oxygen, and you've got that electron, so you've got the carbon dioxide and water being formed. With anaerobic respiration, though, you don't have oxygen, so you have another source as the final electron acceptor. Okay. Now, when we're talking about glucose, you've probably carbohydrates. Carbohydrates are where you get the majority of the cell's energy from. So there's 3 different types of carbohydrates. You've got monosaccharides, so single sugar, 2 sugars, a disaccharide, and polysaccharides. So lots of sugars together. And glucose is the most commonly used carbohydrate. It's got 6 carbon-hydrogen bonds, and you've got that on the diagram there. And the 2 major types of breakdown of glucose are aerobic respiration or anaerobic respiration, also known as fermentation. Okay, that's when you get lactic acid being produced. You also get ethanol being produced, etc. So you've got that occurring, but no oxygen. Now, for the breakdown of a carbohydrate, for example glucose, you go through a number of steps. So it's basically boiled down to 4 stages. You've got glycolysis, so occurring in the cytosol, you've got glucose coming in, going into the glycolysis step, and that glycolysis, there's a number of steps that occur that produce acetyl-CoA. And that acetyl-CoA feeds into something known as the Krebs cycle, or the tricarboxylic acid cycle, and produces electrons to go into the electron transport chain. Okay? From that electron transport chain, you get water being produced, but you also get energy. Okay? You can see here that from the Krebs cycle, you also get ATP being produced, and from glycolysis, you also get ATP being produced. So there's a number of steps to it, but you do get production of energy from different ones. So if we look at stage 1, so the glycolysis, it occurs in the cytoplasm in both prokaryotes and eukaryotes, and both aerobically and anaerobically. So it can happen under both oxygen situations. And you get from 1 molecule of glucose, you get 2 molecules of ATP and 2 of NADH. And the end product is 2 pyruvic acid molecules. Okay, so once when you're going through this, I would probably advise just getting a diagram and writing inputs and outcomes. Okay? And this does look a little bit daunting, but you can see in step 1 and 3, you've got 2 molecules of ATP being used. Okay? So you've got the input of energy into the system. Then in step 6, you've got 2 electrons being transferred. Remember that transfer of electrons? Yep. And in step 7 and 10, you've got 2 more molecules of ATP being formed. And then at the end, you've got a total of 4 ATP and 2 pyruvic acid being formed from 1 glucose molecule. So that's what was on the last page. It's just showing you how they were arrived at. But we're dealing with different systems, so there are alternatives to glycolysis. You can have the pentose phosphate pathway, or the PPP, and you can metabolize glucose and other pentoses to generate 1 ATP and 2 NADPH from 1 glucose molecule. So it's just a different pathway that it moves down. And the bacteria that are that usually use this pathway are Bacillus subtilis and E. coli. Okay. There's another Etnodudorov pathway. You've only got 1 ATP being yielded and 2 NADPH as well. This pathway is preferred by Pseudomonas and other Rhizobium, etc. Okay, so it's just another pathway for them to get their energy. Then you've got the formation of acetyl-CoA. And this is basically how, this is the molecule before it goes into the Krebs cycle. And what happens is you get the pyruvic acid, so the pyruvic acid losing a molecule of CO2, so it's decarboxylation, and then it's oxidized and resulting in basically finally in acetyl-CoA. Okay, so you get the production of CO2 here, and then the eventual production of acetyl-CoA. Okay. Then we've got the Krebs cycle. This is the next part. So you've got the acetyl, from the acetyl-CoA, you've got transfer of electrons, and from one molecule of glucose, the oxidation produces 6 molecules of NADH, 2 of FADH, 2 molecules of GTP. Now this is guanosine triphosphate, and you also get 4 molecules of CO2. So you can see where that CO2 is coming from that we get rid of in our respiration. And this is not for you to replicate. This is just to show you how it cycles through that. Okay, so you've got glucose undergoing glycolysis, producing the acetyl-CoA and moving through. Okay, so you've got the acetyl-CoA and it's moving through. Then we have the next step, that electron transport chain. And don't be too worried about this. It's basically, I want you to get from this, I want you to get to know that it's the cycling of electrons. And this occurs in, so those complexes that I showed you in that first section, you've got the flavoproteins, cytochromes and ubiquinones. They're the ones that transfer the electrons. Okay, so remember those complexes that I showed you? And this electron transport chain occurs in the plasma membrane of prokaryotes and just inside the mitochondrial membrane in photosynthetic. Sorry. Okay. And yeah, so inside the inner mitochondrial membrane in photosynthetic membranes. And basically, this is a way of getting the energy through. Okay, so moving electrons and moving protons and then having oxygen as that final electron acceptor, and then because that happens, then you get the production of ATP. Okay, which is a really good thing. So, why would we be talking about this? Well, you need energy for the cell. You need that ATP to be produced. Okay, there's a number of different things that you need that energy for in the cell. And if something blocks that energy, so it's not just prokaryotes, it's eukaryotes as well that have that electron transport chain, because every cell needs energy, yes? So, have you heard of cyanide? Yes? Cyanide actually blocks this part here. So it blocks that final complex, and so you don't have that transfer of electrons to that last acceptor, to the oxygen, and therefore, if that doesn't happen, then you don't get ATP being produced. And what happens to a cell that doesn't have ATP being produced? It dies. Okay, so that's why cyanide is such a potent poison, because it blocks that electron transport chain. Now, another term that we used was chemiosmosis. Chemiosmosis just means that it's the movement of ions across a membrane down the concentration gradient. So it's kind of like osmosis with water, but in this one, it's the movement of ions, so chemicals across or down their concentration gradients. And proteins being pumped across a membrane generate a proton motive force, a PMF. Now remember in that electron transport chain you had protons being pumped? Yes? So that's needed to create that energy. And that energy is produced when the protons actually move back across that membrane, and they help with the formation of ATP. Everyone remember that picture? Do I have to show you again? Okay, so you've got electrons moving, but you've got protons being pumped out, and then you come to this final bit, but the protons go back in, form water, but also cause the production of ATP. So electrons and protons, you have to talk about them together. And that, that force can be used to move things. So if you're talking about a flagella, it can be used to move the flagella. So once again, you've got the electron transport chain, you've got lots of hydrogens coming out. That hydrogen, those hydrogens come back into the cell, allow the production of ATP. That ATP is then used to allow the flagellum to move. Okay, so this is actually quite a nice diagram to bring it all together, because you can see that the movement of the electrons, the movement of the hydrogen ions, but you have an outcome as well. Okay, and once again, the flagella has got that kink in it, so it can move. So that's what chemiosmosis is about, the movement of chemicals or ions. And so if we were going to put that all together, you've got, if we're talking about aerobic respiration, in prokaryotes, you've got 38 ATP molecules can be produced from complete oxidation of one glucose molecule. Okay, so if you go through all of the steps and count them up, you'll have 38 ATP molecules being produced in prokaryotes. But in eukaryotes, it's not as efficient. You only get 36 ATP molecules being produced from one glucose molecule. And you can imagine how many glucose molecules there are in your body from one mouthful of carbohydrates. Yeah, so we've got a lot of cells and they all need energy, and that's what's happening. And so if you look at it in a little bit of a summary, we've got the prokaryotes, because it's got 38 ATP being produced. You can see where they're coming from, and that electron transport chain produces about 34 ATP. So it's quite efficient. And you can see that they also produce quite a bit of waste as well. So that's why it's important to have that, those transport systems so the cells can get rid of waste. Now, if you think about a bacteria growing in a particular medium, and it's non-motile. It can grow quite nicely, it can reproduce, etc., but it's still producing a lot of waste, and it's using a lot of the nutrients in that environment. Agreed? So if it's non-motile, it's going to stop growing after a while when it's exhausted the nutrients, or the waste concentration is too high. But if it's motile, and say it's in a semi-liquid or a liquid environment, it can grow, it can use the nutrients in that environment, it can produce waste, but if that gets a bit too toxic, if it's starting to become hostile, if it's motile, it can move away. Yes? And some of these things you're going to be looking at in prac classes as well, we're going to be looking at growth trajectories under different circumstances. So if bacteria are grown in a lovely nutrient area, environment, they will have a particular growth trajectory. But if that environment is not so accommodating, then their growth trajectory will change quite dramatically. And that's what you guys are going to be looking at in your pracs. So in summary, we've looked at catabolism and anabolism, and you can tell me the difference between that quite easily. Yes? You can give me examples. Yes? You've got different strategies to produce food, so you need that energy source and a carbon source. We've looked a bit at redox reactions, so basically the movement of ions, electrons and protons, and how to produce energy, a cell needs an electron donor, so it needs something to give up an electron, so you can get that, that movement of electrons, so that's cycling. You've got aerobic respiration is a series of redox reactions, so we've looked specific at the different parts of the glucose catabolism, and when that aerobic respiration occurs, aerobic bacteria, so the prokaryotes, can produce 38 molecules of ATP, whereas eukaryotes can only produce 36. Okay, and you've got the definition of chemiosmosis as well. And so, practice exam questions. What do you think? Number one, an oxidative reaction to a molecule will cause it to? Okay, keep that in your mind, your choice. Then, number two, cyanobacteria are photoautotrophs, which means that we can knock two out of that quite quickly, can't you? Everyone got in their mind what their answers would be? Yes? Okay. Were you right? Okay, well, oh goodness. Spoke too quickly? I don't know. Okay, well, we'll leave it there, and I will see you next week. Thank you.